Myostatin is a protein that in humans is encoded by the MSTN gene. It is a member of the TGF-beta signaling family of growth and differentiation factors. Myostatin is produced primarily in skeletal muscle cells, circulates in the blood, and acts on muscle tissue by binding to a cell-bound receptor called the activin type 2 receptor. Also, mice are often the main model organism when studying the effects of myostatin. Muscle atrophy, also known as disuse atrophy, is the decrease in the mass of the muscle. Atrophy can be a partial or a complete wasting away of the muscle. Many factors may contribute to muscle atrophy, including a single or a combination of several common diseases, starvation or malnutrition, or disuse or immobilization. As seen in the figure, atrophied muscles are decreased in size, strength, and mobility. Approximately 1 in every 6,000 individuals suffer from spinal muscular atrophy. However, others may also suffer from cachexia, which is muscle atrophy as a result of several common diseases including cancer and diabetes. In a study done by Zhu in 2000, the effects of myostatin inhibition was studied. In the gastric, tricep, and quad of the mouse muscle, the DNMS mice shown in the left graph, with an absence of myostatin, were found to have a larger muscle weight than in the control mouse. In the graphs on the right, the cross-sectional area of the muscle of a DNMS mouse was 20% larger than the control. In the lower graph, it is seen that the number of myofibers was less per section in DNMS mice than in the control mice due to increased myofiber size in the DNMS mice. Thus, myostatin is hypothesized to be a negative regulator of muscle growth. The purpose of this experiment was to evaluate the use of myostatin inhibition in specific models of acquired muscle atrophy. The hypothesis stated that myostatin inhibition carries therapeutic potential to prevent and/or ameliorate the loss of muscle mass in models of acquired muscle atrophy. The TA, or the tibialis anterior, is a muscle that in humans is mostly located near the shin on the lateral side of the tibia. The TA also exists in mice and was the main muscle analyzed in this study. Morphometric analysis, which analyzed the size and shape of muscle fibers, were conducted. 10 mm sections of tissue from the TA muscle for each mouse model were cut and stained for laminin gamma 1, which stained the border of the fibers. Pictures were taken of the entire muscle section, and computer macro measured the size and number of fibers. About 1,000 fibers for each tissue section were counted. Data measured were total fiber number, area, as well as minimum ferrets diameter, or MFD, which measured the rough diameter of each muscle fiber. There were two main atrophy models used in this study. An immobilization-induced atrophy model was conducted through a surgical staple attached to the lower limb, which restricted the TA, or the tibialis anterior movement. This induced a single limb atrophy. A denervation, or the interruption of the nerve connection to the muscle, induced atrophy was conducted through the removal of the sciatic nerve from one leg. This denervated the entire lower leg compartment, also inducing a single limb atrophy. Three groups are involved in the study: the control, the immobilized atrophy model, and the immobilized atrophy model plus ACT2B, which is the myostatin inhibitor. The total body weight was measured, and a 10 mm TA section was cut from each mouse. The TA mass was measured, and a morphometric analysis was conducted, which measured the area, total fiber number, and MFD. Statistical analysis was run on the morphometric analysis. A similar methodology was conducted on the denervation phase, except for the groups which included a control, a denervation atrophy model, and a denervation atrophy model plus ACT2B. This graph shows the total change in body mass in the control, immobilized, and immobilized plus ACT2B mice. Since myostatin is well conserved across the entire organism body, there is a total change in body mass with an inhibition of myostatin, as seen in the significantly higher body mass that existed in the immobilized plus ACT2B, or the myostatin-inhibited mice, than in both the control and the immobilized. However, there was no significant difference shown between the control and the immobilized. This figure shows the mass of the TA in the immobilization phase. As shown in the figure, the TA of the immobilized is significantly lower than both the control and the myostatin-inhibited mice. There's also no significant difference between the control and the immobilized plus ACT2B, suggesting that the myostatin inhibition rescued the atrophy. These images represent the laminin gamma 1 stained fibers of the control, the immobilized, and the immobilized plus ACT2B. There is a visual difference between the immobilized and both the control and the immobilized plus ACT2B fibers. This graph shows the total body mass in the control, the denervated, and the denervated plus ACT2B mice. Again, since myostatin is conserved across the entire species, the denervated plus ACT2B, or the myostatin-inhibited mice, have a higher total body mass than the control and the denervated mice. This graph shows the TA mass in the three groups in the denervation phase. Despite the higher total body mass in the denervated plus ACT2B mice, or the myostatin-inhibited mice, the control had a significantly higher TA mass than both the denervated and the myostatin-inhibited mice. There's also no significant difference between the denervated and the denervated plus ACT2B TA, suggesting that in this instance, myostatin did not rescue the atrophy from denervation. These images show the laminin gamma-1 stained TA muscle fibers in the denervation phase. As seen in the pictures, there is no visual difference between the denervated and the denervated plus ACT2B, but the control muscle fibers are much larger than both of the others. Although myostatin specifically is characterized only to exist in vertebrates, some applications may be possible in the lab. This study done by Lowe in 1999 showed that myoglyanin was a gene in Drosophila that was most closely related to myostatin in vertebrate organisms. In C. elegans, the UNC129 and the DAF7 gene have been shown to be partly homologous with myostatin in vertebrates. This is my works cited, and that concludes my presentation. Thank you.